Hello, everyone, and welcome to this Genetic Engineering and Biotechnology News webinar. It's entitled Current Trends in 3D and Organoid Cell Culture for Cancer Research. 2D cell culture has played an important role in advancing life science research in areas such as developmental biology, cancer research, and drug discovery. But this methodology cannot reflect the complex cellular architecture and structure found in the in vivo environment. A better approach was needed, and that has come in the form of 3D cell culture technology. What's more, these cell culture systems are especially applicable to the study of cancer. They can be used to grow organoids that can replicate critical properties of in vivo organ systems or the original tumors from which they were derived. And that's the focus of today's webinar. I'm John Sterling, Editor-in-Chief of GEN, and I'm going to serve as the moderator. But first, let's meet our panelists. Dr. Marshall Kozovsky is Global Scientific Support Manager for Corning Life Sciences. He will discuss Corning's materials and systems for optimizing 3D cell culture environments. Wojciech Senkowski, a doctoral candidate at Uppsala University, is going to talk about spheroid-based high-throughput drug screening. And Dr. Omer Yilmaz serves as an assistant biology professor at MIT. Omer will describe the application of ex vivo intestinal organoid assays to research on the dietary control of stem cells in physiology and cancer. After the panelists make their presentations, there will be a question and answer segment. Feel free to send in a question for our panelists at any time during the webinar. Type your question into the Ask a Question box on the left-hand side of your console and then hit Submit. The panel will try to answer as many questions as possible. So, if everyone's ready, let's get going. Dr. Marshall Kozovsky will be our lead-off presenter. Marshall?
Thank you, John. Hello, and thank you for joining us. Today, we will hear talks about two different 3D cell culture systems that model in vivo cellular behavior. Such models are being used increasingly for drug discovery and to study the biology of tissues and diseases. In relation to these talks, I will give a brief overview of Corning Life Sciences products that are suitable for 3D cell culture, including the generation of cell spheroids and organoids. Corning offers a broad variety of animal-derived and synthetic matrices, including Matrigel matrix, laminin intactin complex, collagens, and Corning Puramatrix peptide hydrogel. These materials have been widely used to support differentiated cell morphology and function in 3D cell culture environments. Corning spheroid microplates enable the generation of cell spheroids from a variety of cell types. And Corning permeable supports, or cell culture inserts, provide a dynamic platform that can be used to establish complex 3D culture models that incorporate multiple cell types. This slide shows two examples of 3D matrices that support cell differentiation and functionality. On the left, mammary epithelial cells exhibit differentiated morphology, marker expression, and polarized structure in a 3D overlay system comprised of Matrigel matrix. The image on the right shows a rat primary hepatocyte spheroid on a 3D matrix comprised of Corning Puramatrix peptide hydrogel, a synthetic, animal-free biomaterial. Corning spheroid microplates are multi-well black clear plates with a round bottom, which is treated with the Corning ultra-low attachment, or ULA coating. The unique design of the well bottom, in conjunction with the ULA coating, enables the generation of reproducible and well-defined spheroids from a variety of cell types, including tumor cells, stem cells, and primary cells. Spheroid formation can be performed in 96 or 384 well plates. Each well generates a single spheroid, which is amenable to direct analysis in the same plate using fluorescence or luminescence detection. In this experiment, human neural stem cells were cultured in the Corning spheroid microplate. These cells assembled into neurospheres, which were shown to exhibit multipotency and the capacity to differentiate into neurons, oligodendrocytes, and astrocytes. Corning permeable supports are comprised of two compartments separated by a microporous membrane. These systems can be used to create unique and complex 3D culture environments. This schematic depicts various options for establishing a 3D co-culture model. Cells can be seated onto three different surfaces: the top of the membrane, the bottom of the membrane, and the floor of the well. In addition, as depicted by the red stripes, surfaces can be coated with 3D matrices to further define the growth environment. These options enable a variety of conditions, such as the physical separation of different cell types, paracrine signaling, and in some cases, the physical interaction of cells through the pores of the membrane. In summary, Corning offers a variety of materials suitable for 3D culture, including ECMs and an animal-free peptide hydrogel. The Corning spheroid microplates provide a novel platform for generating defined spheroids for basic research and drug discovery. And finally, I've discussed the use of permeable supports for establishing a dynamic 3D cell culture system in a two-compartment environment. Today, you will hear two talks that demonstrate the utility of 3D cell culture, one using tumor spheroids to more accurately screen for anti-cancer compounds, and the second focusing on the generation and analysis of gut organoids in the context of diet and cancer. Thank you again for joining us today. If you have any questions about the Corning products or applications that I've described today, please contact us using the information provided at the end of this event.
Thank you, Marshall, for that overview of the types of materials that are available for 3D cell culture activities, and also for how to set up a 3D cell culture environment. You definitely set the stage for the talks that follow, so thanks again. If you're just now joining our webinar, thanks for tuning in. Just a reminder that there will be a question and answer segment after the last presentation. Please put your question into the box on the left-hand side of your console and then hit submit. Our next speaker is Wojciech Zankowski. Wojciech.
Thank you, John, for your kind introduction, and thanks to Corning for inviting me to speak to you today. But right before I jump to high-throughput drug screening assays that we have developed and applied in our lab, let me just ask the question, why would we even need spheroids for high-throughput drug screening? So on the left side, we see a standard monolayer cell culture, the model that is most widely used in cancer research and drug development today. However, what we are usually up against looks quite different, like this tumor on the right. Now, if we take a glimpse right into the tumor microenvironment, we can see that this is very heterogeneous. Here we can see a staining for K67, a marker of cellular proliferation. We can see in this picture that there are not many proliferating cells, and they are mostly concentrated around what I think is a blood vessel. However, if we perform the same kind of staining in monolayer cell culture, we can see that virtually all the cells are proliferating. What we lack in monolayer cell culture is the part in the circle. This is the part where cells are quiescent. The cells are not dividing, and this is the part that we would like to simulate with spheroids. This is also the part that is quite drug resistant because standard chemotherapy works mostly on proliferating cells. Since the cells in these quiescent areas are not dividing, they are quite resistant to the therapy. Secondly, they also are quite far from blood vessels, so drugs have a quite hard time even reaching those cells. This is quite apparent when we take a look at this fluorescent microphotograph. With a red color here, we have marked blood vessels. With green color, hypoxic areas where quiescent cells reside. These are the cells of interest that we want to target. And with blue color, we see doxorubicin. Doxorubicin is a standard chemotherapeutic drug. As we can see already from this picture, is that doxorubicin doesn't really penetrate very far into the tumor tissue. So when you think about our model, first we would like to have a model that would comprise quiescent cells, but also that would help us evaluate the penetrative properties of investigated compounds. And to our aid comes a spheroid model, like presented on the picture. Here we can see a spheroid of around a few hundred micrometer in diameter. Spheroids usually experience the gradient of oxygen pressure that results in internal hypoxia. And the same situation comes for nutrients, which results in low cellular proliferation inside of the spheroid and only a few proliferating cells concentrated on the periphery of the spheroid. And the same situation applies when we think about drugs. So we have the gradient of drug concentration if we apply treatment to our spheroids. And this is even more apparent when we take a look at the staining for caspase-3 performed on the spheroid treated with cisplatin, a standard antiproliferative drug. So the cells that are positively stained, the cells that are apoptotic here, that are dying, are only concentrated on the very peripheral part of the spheroid. So the drug didn't penetrate very far into the spheroid tissue, or it just didn't work on the cells inside of the spheroid. Now we can compare monolayer cell cultures and spheroids in dose response experiment, and we see that spheroids are way more resistant to a standard chemotherapeutic drug, mitomycin. As Marshall already mentioned in his introduction, spheroids can be formed from a wide range of different cell lines, but in this presentation I will mainly present data from HCT116 spheroids. These are spheroids formed with colorectal cancer cell line. For high-throughput drug screening, we need to form spheroids in a high-throughput format, meaning at least 384 well format. And that was quite problematic a few years back to obtain same size uniform spheroids and only one single spheroid in each well. What we had at the time were only ultra-low attachment cover plates, but these were flat bottom plates. So we had to come up with an idea to form the desired spheroids in those plates. And the way we did that was that after cell seeding, we incubated our plates in what we called a tilted position. So one corner of the plate was lower than the other corners of the plate. Now, when we take a look at the schematic representation of the inside of the well, we can see that cells aggregate in the corner of the well. Then we placed our plates on a laboratory rocker, but not lying flat on the laboratory rocker, but instead we placed the plate standing on the edge. So the angle between the laboratory rocker shelf and the bottom of the plate was around 85 to 90 degrees. And then we slowly rocked the plate every 3 hours for 15 minutes. So if we take a look at the inside of the well and what is actually happening to our cell aggregate, we can see that it's getting more and more round, like a snowball. After this step, we place the plates back in the tilted position and we let the spheroids grow for the rest of the culture period. Right now, the spheroid formation process is much easier when we have the U-bottom plates from Corning. And after cell seeding, we just need to shortly centrifuge the plates to induce the cell aggregation, and afterwards we just let the spheroids be for the rest of the week, and we just grow them in the incubator. There's an important variation in our spheroid culture process that is different from the common, most widely used spheroid culture method. And this variation is that we do not exchange the culture medium over the whole spheroid culture period. So when we take a look at the glucose concentration and pH in the culturing medium, we can see that they both gradually decrease. And as you probably remember, we wanted to simulate the quiescent part of the tumor and the conditions that are present in there. So we wanted to add our drugs at the time point when the glucose concentration and pH start reaching this in vivo based values. And this is why we decided to add our drugs after 7 days of the spheroid formation. What we also need for a successful high-throughput drug screening is a good readout assay, and we wanted to know when our spheroids die during the treatment. So for that, we used HCT116 cells that constitutively expressed GFP, green fluorescent protein. So when cells die and the protein translation ceases, the spheroids lose their fluorescence. Now we can couple that to the automated fluorescent microscope, and we can very fast and very efficiently measure the whole plate of spheroids. Now we can see the differences between treated and non-treated spheroids very easily. And when we take a look at the numerical values, we can see that the system is very repeatable and quite well suited for high-throughput drug screening. This method also performs very well in the dose-response experiments, and since we do not add any external reagent to wells, we can monitor the viability of the spheroids over time and measure the plates basically as many times as we want. So here you can see the measurement after 48 hours, 72 hours, and 96 hours after drug addition. These are the same spheroids. So armed with all these assays, we performed the screen of a library consisting of 1,600 compounds. We basically added them to spheroids, and we chose 41 that were the most potent compounds in killing our spheroids. Then we tested these 41 compounds in a monolayer and spheroid-based dose-response experiments, and we chose 12 which were more potent in killing spheroids than monolayer cell cultures. We call them 3D selective. However, as you probably remember, we used the GFP assay for this. However, GFP assay doesn't really show when cells die. It just shows when cells stop translating proteins. So we wanted to check if our hits really kill spheroids, and for that we performed 3D-based clonogenic assay. What we basically did was that we added drugs to our spheroids for 72 hours at the 2D-based IC50 concentration, the concentration that killed off 50% of cells in monolayer cell cultures. After the treatment period, we dispersed the spheroids into single-cell suspensions and seeded the cells in the fresh media into 6 well plates. We let them regrow for 10 days and then we stained the colonies. So when we take a look at the results, there's quite some regrowth from the spheroids that were not treated with anything, and still quite some regrowth from the spheroids that were treated with doxorubicin or oxaliplatin, standard chemotherapeutic drugs. However, there were 5 drugs that abrogated the colony formation completely. And out of these 5 drugs, we chose one, nitazoxanide, as the most interesting one, because this is an FDA-approved antiparasitic agent that reaches very high plasma concentrations with virtually no side effects. If we go back for a second to our 5 final hits, we noticed that they share, that they target the same mechanism, and the mechanism was mitochondrial respiration, also known as oxidative phosphorylation, the process in which most of cells' ATP is produced. 3 of 5 compounds were uncouplers of mitochondrial membrane potential, meaning that they freely transfer protons across the mitochondrial membrane, so they abrogate the gradient of the protons across the membrane, and cells cannot use this gradient to produce ATP. When we take a look at the structures of these compounds, we can see that all the uncouplers share the same pharmacophore. Now we wanted to validate and characterize the effects on mitochondria. And what you can see on the left is the direct comparison of nitazoxanide and FCCP, a standard, well-known uncoupler. So what we measured here was oxygen consumption rate, the speed at which cells consume oxygen. So the profiles of two drugs look very similar, and when we apply a low concentration of the drug, we see a boost in oxygen consumption rate. However, when we increase the dose of the drug, this period of elevated oxygen consumption gets shorter and is followed by shutdown of mitochondrial respiration. And all of this was performed in monolayer cell cultures. However, in spheroids, we see a very, very similar thing. Here we stained for hypoxic areas, and after a short-term treatment with nitazoxanide or a standard uncoupler CCCP, we can see that cells consume more oxygen because the hypoxic areas are quite large. However, after the long-term treatment, the hypoxic areas are gone, so the mitochondrial respiration has stopped. And the final confirmation comes from the staining with JC1 probe. This is a fluorescent probe that positively stains polarized mitochondria, and in both CCCP and nitazoxanide-treated wells, we don't see any red fluorescence from the probe, we just see the blue fluorescence from the cell nuclei. However, one has to remember that we chose nitazoxanide to target only the quiescent part of the tumor. So we wanted to combine it with something that would target proliferating cells, and the easy answer would be the anti-proliferative chemotherapy. So what you can see here is the direct comparison of drug effects on monolayer cell cultures and spheroids. When we apply oxaliplatin, the standard chemotherapeutic drug, we can see a strong cytostatic effect on 2D cultures, the monolayer cell cultures. However, spheroids are largely unaffected over 72 hours of treatment. With nitazoxanide, we see quite opposite situation. The spheroid dies gradually, and monolayer cell cultures have basically no problems proliferating. So we speculated that the approach of combining nitazoxanide and a standard chemotherapeutic drug might show benefits in an in vivo model, and this is exactly what we saw in the mouse xenograft model when we combined nitazoxanide and another chemotherapeutic drug, irinotecan. Nitazoxanide here potentiated the effect of irinotecan. Sometimes, instead of using a quiescent spheroid model, we might want to use a more proliferative one, the one that is not so metabolically stressed. And for that, we need to exchange the culturing medium during our spheroid culturing process. So after the cell seeding and centrifugation and 4 days of spheroid growth, when the medium is digested, we first aspirate it with a 96-channel microplate washer. Then we add a fresh medium and centrifuge the spheroids to place them back on the bottom of the well. Then we repeat the whole process, first aspirating the medium, then we add the fresh medium again, and then we centrifuge the spheroids to place them back on the bottom of the well. And in this way, around 97% of old medium is replaced with the fresh one. And this approach results in the generation of 3 different cellular models from the same cell line. So we have a standard monolayer cell culture model. Then we have the most commonly used proliferative spheroids, as we call them, the spheroids that were formed with the medium exchange over the culture period. And then we have our quiescent spheroids that were formed without medium exchange. So both spheroid models actually show a quite vast hypoxia. I don't have it in the picture right here, but so you have to believe me. But what is unique about quiescent spheroids is the low physiological glucose concentration. Now we can ask if the medium exchange, just the medium exchange over the culture period, influences the response to the treatment of the spheroids, and the answer is yes. And it influences it quite dramatically. When we apply our 5 OXPHOS inhibitors, we can see that spheroids formed with medium exchange are much more resistant to all those inhibitors. And this data are based on GFP assay, but we see the same and even more dramatic effect from the clonogenic assay. And this is all I have today for you. And with that, I would like to summarize with a few take-home messages. I would first like to draw your attention to the GFP-based assay of measuring spheroid viability, because this is a very good and non-invasive marker of the viability, and that's why this is well suited for high-throughput drug screening. Then I would also like to draw your attention to the target here, oxidative phosphorylation, that has emerged in many papers over the last, I would say, 2 years as a very promising target in quiescent and nutrient-deprived cells, in the cells that are the most problematic to treat. And finally, I would like you also to remember that when we culture something to perform drug tests or simple cell biology experiments, we always need to remember about our cellular model that we are using and the conditions that we are using in our culture, because as I showed you before, the differences in the response to the treatment can be quite dramatic. And the results I presented are the effort of quite many people, and so I would like to just say a few thanks. Thanks to my group and my panel of supervisors from Uppsala University, especially Morten, who is a person who I work mostly on spheroids, but also thanks to the group from Karolinska Institute, who performed a lot of oxygen consumption rate measurements for us and other assays as well. And with that, I would like to also thank you all for listening today, and I'll be happy to take any questions either in the Q&A that follows this presentation, or you can contact me directly via email. So thank you, and back to you, John.
Great job, Wojciech. Thanks for demonstrating how GFP fluorescence can be used to determine spheroid viability for high-throughput screening, and also for talking about how culture conditions dictate treatment outcome. Just a reminder that there will be a question and answer segment after the next presentation. Please put your question into the box on the left-hand side of your console and then hit submit. Dr. Ömer Yilmaz is all set and ready to go. Ömer, the floor is yours.
Thanks, John, for the introduction. So today I'm going to talk about work in our lab which deals with dietary control of stem cells in physiology and disease. So the question that my lab is interested in addressing is, how does organismal diet impact stem cell function in physiology and disease? And we interrogate this question by looking at diverse diets. On one side of the spectrum, my lab is really interested in understanding the effects of low-calorie diets such as caloric restriction, which is this really interesting phenomenon where when one cuts their calorie intake by 20 to 40%, this has positive effects on many parameters of health, and in some cases on organismal lifespan. In addition to calorie restriction, we look at short-term food deprivation and short-term fasting. On the other side of the spectrum, we're interested in looking at conditions associated with overeating and an overabundance of nutrients, such as a high-fat diet, Western diet, and genetic models of obesity. And we think that these types of models are very germane for the diets that comprise Western civilization. And lastly, we're really interested in studying these effects of diet because we know that diet has strong effects on aging and diseases such as cancer. So we really want to gain insights into how different diets regulate pathways and try to understand the biology of those pathways and their impact on diseases of aging. So an example of this would be obesity, where obesity is a diet modifiable risk factor for cancer. So in obesity, for example, we know that it correlates to increased cancer incidence in many tissues. And relevant for today's talk, there's epidemiologic data linking a body mass index of greater than 30 with a modest increase in colon cancer in both men and women. So our tissue for understanding the effects of a pro-obesity diet on stem cell function and tumor initiation is the mammalian intestine. In the mammalian intestine, we know that there are intestinal stem cells which reside at the base of these intestinal crypts shown on the left here in schematic. So highlighted in dark green are the intestinal stem cells, and thanks to the pioneering work of Hans Clevers and colleagues, we know that a majority of the intestinal stem cells in mammals express a gene called LGR5, and in mice, using a genetic approach, we can knock in a green fluorescent protein into the promoter of that gene and identify these LGR5 GFP positive cells as intestinal stem cells. These LGR5 GFP positive stem cells give rise to transit amplifying daughter progenitor cells, and these transit amplifying progenitor cells then give rise to the various differentiated cell types of the intestinal epithelium. And in the mouse intestine here shown in schematic, these differentiated cell types are in these finger-like projections called villi of the small intestine. So in the intestine, these LGR5 positive cells are also adjacent to these panel cells, which comprise an important component of their stem cell niche or stem cell neighborhood. And these panel cells elaborate key factors that regulate the biology of these stem cells. So in the intestine, like many adult tissues, one has a very rare population of stem cells, which in the intestine, this population is LGR5 positive stem cells. And this rare population gives rise to the majority of differentiated cells, which in the intestine are LGR5 negative and include the absorptive cells as well as all of the secretory cell lineages. The intestine is a great system to study, not only because we know the identity of these stem cells and where they reside within the intestine, but also because one has very robust functional assays to interrogate how different dietary interventions regulate their function in ex vivo assays. So for example, one can use flow cytometry to isolate the LGR5 positive stem cells and their Pannis niche cells from different dietary conditions, co-culture them in matrigel, and in the presence of appropriate growth factors, these stem cells give rise to these clonal mini-intestines or organoid bodies in culture. And these organoid bodies have a morphology that's reminiscent of the morphology that one observes in the in vivo mammalian intestine, where they possess these cryptomains and these villus domains. And on top here is a fluorescent image from an organoid derived from an LGR5 positive stem cell, and one can appreciate that these LGR5 positive stem cells reside at the base of these intestinal crypts in these organoid bodies, consistent with where they reside in vivo in the mammalian intestine. So my lab is really interested in testing how different dietary constituents in this assay alter stem cell function. So to understand how diet-induced obesity regulates intestinal stem cell function in our model organism, which is mice, we place mice on a long-term high-fat diet. And just like has been observed in human patients, this high-fat diet-mediated obesity leads to an increase in spontaneous intestinal tumor formation in our model organism. So here one can appreciate that on a long-term high-fat diet, these mice become quite obese, and these mice develop spontaneous intestinal tumors here, shown grossly and microscopically, and these mice, shown in the red box, have a 3 to 4-fold, are 3 or 4-fold more likely to develop invasive intestinal tumors. So we wanted to understand how this high-fat diet alters the biology of these intestinal stem cells. So we performed many assays, one of which I'm showing here, and asked, how does a high-fat diet change the numbers of intestinal stem cells in these mice? And as one can appreciate here in the bar graphs, there's about a 2-fold increase in the frequency of these primitive GFP high stem cells in a long-term high-fat diet, indicated by the dark bars, whereas there's no significant change in the frequency of the more differentiated progenitor cells, which are GFP low. And these data reflect what we find in the small intestine, and data that I'm not showing you, we observe similar fold changes in the colon. And we validated these findings using many independent techniques, such as in situ hybridization, using alternate markers for intestinal stem cell identity, such as olfactomedin 4. So we then wanted to ask, in addition to quantitatively changing stem cell numbers, does a high-fat diet change the quality or the function of these intestinal stem cells? So to do this, we again used our organoid assay to interrogate how a high-fat diet altered the function of these intestinal stem cells in the organoid assay. So here we're looking at the ability of a GFP high stem cell to give rise to a mini intestine in culture, and on the horizontal axis here, we're looking at the dietary conditions from which stem cells and their niche pana cells are derived. So here we see that control stem cells by themselves are ineffective at giving rise to these mini intestines in culture, and this is also shown pictorially in the upper right corner. When one cold cultures a control stem cell with a control pana cell, the presence of a pana cell dramatically enhances the potential of the stem cell to give rise to a mini intestine in culture, accentuating the fact that these niche cells play an important role in regulating stem cell function. Interestingly, and really surprisingly, we find that these intestinal stem cells that are derived from a high-fat diet, these stem cells are poised for niche-independent growth. And these stem cells are as clonogenic as control stem cells that are co-cultured with control pana cells. Moreover, these high-fat diet stem cells can still receive and respond to cues that are derived from the control pana cell niche. We think these findings are really exciting because they indicate that a high-fat diet primes stem cells for niche-independent growth. And this raises the possibility that maybe an early change that occurs in the intestine with a long-term high-fat diet is that these stem cells become uncoupled from their neighboring niche cells. So we then turned our attention to the diet itself, and we wanted to understand, do constituents of the high-fat diet, such as palmitic acid, mimic the effects of a high-fat diet on intestinal stem cells? So just to give you a little bit of background on this high-fat diet is comprised of 60% fats, and the main component of this high-fat diet is lard. And the main constituents of this lard are saturated fatty acids such as palmitic acid and unsaturated fatty acids such as oleic acid. So we asked a very simple question. Just as a high-fat diet increases stem cell self-renewal, where a high-fat diet leads to more intestinal stem cells, we asked, what effect does palmitic acid have on intestinal stem cell numbers and function? So to test the effects of palmitic acid on intestinal stem cell numbers and function, we took normal crypts from LGR5 GFP reporter mice, and we grew them either in the presence of vehicle or palmitic acid here, shown on the left. And one can appreciate even by immunofluorescence that by co-culturing these intestinal crypts in the presence of palmitic acid leads to an increase in greenness. And remember, green is a proxy for the numbers of LGR5 positive cells. We looked at hundreds of crypts using flow cytometry, where we can harvest many wells of our organoids growing in matrigel and quantitate in a high throughput manner using flow cytometry the frequency of GFP. And one finds that just as we observed by immunofluorescence, there's about a twofold increase in the frequency of GFP high intestinal stem cells in these organoids that are co-cultured with palmitic acid. And lastly, not only does palmitic acid lead to an increase in the frequency of LGR5 positive stem cells, it also confers on them the ability to grow and initiate organoids independent of their niche cell. And that's shown on the right, where we're looking at organoid per stem cell from vehicle-treated organoids and palmitic acid-treated organoids. And one can see that stem cells from palmitic acid-treated organoids are 3- to 4-fold more likely to initiate these mini-intestines. We don't think that these effects are specific to palmitic acid because we observe similar effects with other fatty acids that comprise the high-fat diet, such as oleic acid. We've used commercial mixtures of lipids, and we don't think these effects are specific to mice because we're able to mimic them in human-derived organoids. So an important question is, what is the mechanism mediating these effects of a high-fat diet and fatty acids in culture? So to address this question of mechanism, we performed RNA sequencing on intestinal stem and progenitor cells from high-fat diet mice and from control chow-fed mice. And one pathway in particular really jumped out at us upon analyzing the data, and that is the PPAR pathway. So these PPAR family members are nuclear receptors, and upon engagement of endogenous lipid signals, they engage a robust metabolic program. And in this case, we think our main PPAR family members is a protein called PPAR delta, and PPAR delta upon activation leads to robust activation of fatty acid oxidation, which enables cells to use fatty acids to generate energy in the form of ATP. So in data that I'm not showing you, we've gone on to validate that indeed PPAR delta program is induced robustly in intestinal stem and progenitor cells in a high-fat diet. So we then asked a simple question of whether PPAR delta is necessary, sufficient, or both for mimicking the effects of a high-fat diet. That is, we know that a high-fat diet leads to increased stem cell function and to niche-independent growth. What role does PPAR delta play in driving these effects of a high-fat diet? So to address this, we first used a pharmacologic agent called GW501516. So this GW compound is a potent synthetic ligand that strongly activates PPAR delta signaling. So to test whether PPAR delta activation is sufficient for mimicking the effects of a high-fat diet, we administered this compound to mice for 3 to 4 weeks and assayed its effects on intestinal stem cell biology. So we find that administration of GW in vivo in data that I'm not showing you leads to an increase in stem cell numbers. But not only does it lead to an increase in stem cell numbers, just like a high-fat diet, it functionally enables these stem cells to give rise to mini-intestines in culture without co-culture with their niche cells. So here we're looking at organoid per plated stem cell, and one can appreciate that stem cells derived from mice treated with the GW agonist are several-fold more likely to give rise to these mini-intestines shown pictorially on the right side, where we're looking at day 12 organoids from control stem cells and from animals treated with GW. So these data lead us to the conclusion that GW administration enables niche-independent organoid formation. So as I mentioned to you before, we see very similar effects in primary human mini intestines. So here we grew these mini intestines in Matrigel from patients, and we either exposed them to lipids, palmitic acid, or GW, shown in the bar graph. And just as we observe in our mouse cultures, we find that exposure to lipids, palmitic acid, or to GW, which is the PPAR delta agonist, exposure to these compounds for 2 weeks boosts the clonogenic function of these human organoids in our ex vivo organoid assay. So up until now, I've shown you that a PPAR delta program is sufficient for mimicking the effects of a high-fat diet and its response to fatty acids. But what about necessity? So to address the question of necessity, we deleted PPAR delta in our organoid assay and tested the effect of palmitic acid, lipids, and our GW compound on both PPAR delta wild-type organoids, here shown in gray, as well as PPAR delta knockout organoids, here shown in black. And what we find is quite interesting. We show that when we delete PPAR delta, we now abrogate the effects of this organoid response to palmitic acid, lipid, and to the PPAR delta agonist GW501516, indicating that PPAR delta drives fatty acid-driven organoid self-renewal. So why is this important? If you remember, we embarked on this project to gain insight into why diet-induced obesity correlates to more tumor incidence in humans. So what are the potential contributors in our findings to cancer incidence? So here on the left side, in a normal diet, we know that many tissues are maintained by stem cells shown in red, and these stem cells often accumulate the initial mutations and give rise to cells which undergo oncogenic transformation and give rise to tumors. So what happens in obesity or diet-induced obesity? So what we find in the intestine, at least, is with diet-induced obesity, there's an increase in stem cells here shown in red, so there's more red circles. And because you have more stem cells, one has more substrate that can undergo oncogenic transformation, which can give rise to more tumors. So we think one potential contributor to obesity is that with a high-fat diet, there's an increase in the stem cell pool. So you have more substrate that can undergo oncogenic transformation. We also hypothesize that in diet-induced obesity, perhaps non-stem cell populations can undergo oncogenic transformation and contribute to cancer initiation. So why do we think that? So we think that's the case for because of the data on this slide. So up until now, I've really focused on the effects of a high-fat diet on intestinal stem cells and have avoided talking about the effects of a high-fat diet on their daughter, more differentiated progenitor cells. So here we're looking at the organoid-forming capacity of these GFP-low progenitor non-stem cells in the intestine. So on the horizontal axis, we're looking at the different conditions from which these progenitor cells are derived, and one notices that these progenitors from a high-fat diet or from mice that are treated with the GW PPAR delta agonist, that these progenitors are much more clonogenic in culture. So they're much more likely to give rise to these mini intestines in culture than their control or vehicle counterparts. And this is shown in pictures to the right, where control and vehicle progenitors are unable to give rise to these mini intestines, whereas these high-fat diet or GW progenitors are able to give rise to these mini intestines. And this is exciting because it suggests that a high-fat diet or activation of PPAR delta confers organoid initiating capacity to non-stem cells. Remember, the ability to form organoids is something that we normally ascribe functionally to stem cells. So why is this important in terms of cancer initiation? So we wanted to formally test that these non-stem cell progenitors with PPAR delta activation can indeed initiate tumors in the in vivo setting. So to do this, we deleted a tumor suppressor gene APC, which is often mutated in human colon cancer. In fact, deletion of APC or dysregulation of the pathway that APC controls is dysregulated in up to 90 to 95% of all colon cancer tumors. So we performed a very simple experiment. We treated mice with our PPAR delta agonist and then deleted APC in stem cells and in progenitor cells and transplanted these cells using colonoscopy back into the colon of recipient mice. And the data from that experiment are shown here. So on the left side, we're looking at the transplantation of vehicle stem cells and vehicle progenitor cells that have undergone deletion for APC. And as one can appreciate, only the vehicle-treated stem cells that have lost APC are able to initiate tumors in the recipient colon. And this is consistent with our notion that that stem cells are often the cell of origin for intestinal premalignant adenomatous lesions. In contrast to this, when we delete APC in stem and progenitor cells that have PPAR delta activated with our GW agonist, we find that both stem cells from these GW-treated mice and progenitor cells are able to initiate tumors. And this is quantitated in the bar graph to the right, where stem cells from both vehicle and GW-treated animals are able to effectively initiate tumors in recipient mice, but only progenitors from the mice that are treated with GW are able to initiate tumors, indicating that PPAR delta activation permits APC null intestinal progenitors to form in vivo premalignant adenomas. So my lab is very interested in now exploiting these findings therapeutically. So what are the therapeutic implications of the story that I just described? So my lab is very interested in targeting PPAR delta and its downstream effectors in the appropriate dietary or metabolic context. So for example, today I've shown you that a fat-rich diet or mimetics of a fat-rich diet, such as this PPAR delta agonist, drives PPAR delta activity. This leads to an increase in both stem cell numbers and functions, as well as in progenitor function. And this PPAR delta and this activation of PPAR delta in stem cells and progenitor cells contribute to this increase in intestinal initiation, in this increase in intestinal cancer initiation that we observe in the high-fat diet. So this really raises the question of whether inhibiting PPAR delta in the appropriate dietary condition might mitigate some of these effects that we observe in a high-fat diet or with pharmacologic activation of PPAR delta. And I just want to leave you with two key points from my presentation today, which is stem cells gain niche independence in a high-fat diet, and non-stem cells acquire stem cell properties, such as the ability to form organoids and the capability of undergoing oncogenic transformation in a high-fat diet. And with that, I'd be happy to take questions. So I'd like to acknowledge members of my lab that contributed to the project that I described. Much of the data that I showed you was generated by 3 individuals, a graduate student, Samir Beaz, and 2 talented postdoctoral fellows, Mieko Mana and Jayton Roper, who really pioneered and optimized our ability to transplant these APC null stem and progenitor cells into the colon of recipient mice. And I'd like to thank our various collaborators in the Boston community and from the Massachusetts General Hospital. And with that, I'd like to take any questions.
Thank you, Omer, and thanks again to Marshall and Wojciech for your presentations. You all provided some very valuable insights on 3D and organoid cell culture for cancer research. If the audience would like to know more about Corning Life Sciences products and technologies discussed in this webinar, here are several email addresses where you can get more information. Well, our webinar speakers certainly connected with our audience based on the questions we've received for our Q&A. So let's take the first one.
All right, guys, so the first question is for Wojciech. Wojciech, one of our audience members would like to know which is better, flat bottom or U-bottom? Wojciech, you there? I don't know if he's on mute, or maybe we lost him for a second. Well, we'll get back to Wojciech. Maybe we'll ask Omer, if you're there. We have a few questions for you. One of the first questions one of our audience members would like to know, it said, is it that the diet palmitic acid lead to expression suppression of certain genes in the stem cells?
Yeah, so what we find is when we do the ex vivo culture of these mini intestines with, for example, palmitic acid, just as a high-fat diet activates this really pronounced PPAR delta gene signature, we find that we're able to recapitulate that signature by treating these mini intestines with palmitic acid.
All right, thanks very much, Omer. And I think we're going to ask you another question here. So while Wojciech's figuring out, he's having a little technical difficulty, but he should be back with us for a second. One of our audience members wants to know is, are niche cells formed in the 3D organoids?
Yeah, that's an interesting point. It turned out that, although we find that these stem cells acquire niche independence when initiating these mini intestines in culture, one of the earliest divisions that they do in this matrigel-based organoid assay is that they give rise to their niche cells. So, for example, stem cells from the small intestine early on in culture give rise to their support pana cells.
All right, thanks very much, Omer. I know that Wojciech was trying to call back in. Wojciech, have you got back in yet? No, not yet? All right. So, Omer, we're going to continue with you. We still have a few questions for you. Another audience member would like to know, he says, I'm wondering why you initially decided to use a 60% lard diet. How does this compare to a high carbohydrate diet that is implicated in the development?
Yeah, that's a very good point. We initially wanted to interrogate this question of the effects of a pro-obesity diet on stem cells and cancer initiation using a high-fat diet, but currently in the lab, we're now looking at these high-carbohydrate diets, which are called the Western diet. In addition to these high-carbohydrate diets, we're now looking at genetic models of obesity where our animal models become really obese on just a normal diet. So that is a question we're actively pursuing in the lab.
All right, thanks very much, Omer. I think we have Wojciech back. Wojciech, you there?
Yes, I'm here.
All right, we got you back. So we've got a couple good questions for you. First question for you is, what I started off with was one of the audience members would like to know, which is better, flat bottom or U-bottom?
Well, of course, as I showed you before, our steroid formation process, we had quite complicated formation process in flat bottom plates, and when we acquired the U-bottom plates, our lives got much easier, I would say, because now we just need to seed the cells, then centrifuge the plates, and then we just leave the plates be in the incubator for 1 week. We don't have to do all the rocking steps and changing the position of the plate. So I would say that the U-bottom plates are much better.
Okay, great. Thanks very much. And we'll continue with you since you weren't here for a bit. One of our audience members would like to know, are these outcomes cell line specific, or have you tried other tumor cell types?
Yeah, this is a very good question. We are a bit limited by the number of cell lines that we have that express GFP, but we formed spheroids with a couple of other cell lines, and for example, an HT29 cell line that has a different driver mutation than HTT116 cell line, we see a very similar effect. The 3D selective compounds, the final hit compounds that were killing spheroids more efficiently than monolayer cell cultures, show similar effects in HT29 cell line. And right now we are performing more tests with A549 cell line. This is lung cancer cell line, and I hope one day you will read about it.
All right, thank you very much, Wojciech. And we'll give you one more. One of our audience members wants to know, how is it possible that the cell culture medium is not changed for 7 days of culturing while still having viable cultured cells?
It's a very good question, but you need to remember that cell culture medium has a very high glucose concentration to start with. The McCoy medium that we are using has 17 millimolar glucose concentration, and the most popular medium, DMEM, has the glucose concentration of 25 millimolar. So cells in vivo tumors persist in much lower glucose concentrations, and they are quite fine up until with the concentrations as low as, I would say, 2 millimolar, 2.5. They do not divide in this concentration, but it doesn't mean that they are not alive. They are just quiescent. They stop dividing, and they are just sitting around. But if you put them back into the high glucose concentration, they will start dividing again. So they survive. They just stop dividing.
All righty, thank you very much. We have one question that came in for Marshall. Marshall, the audience member would like to know, does Corning provide protocols for seeding cells on different surfaces of the culture inserts, top and underneath?
Thank you. Yes, so Corning does provide a number of protocols that are available for different application areas such as tumor cell biology, stem cell biology, angiogenesis, just to name a few. In terms of seeding cells onto cell culture inserts, it really would depend on what, if there is a material, what material is on the surface and what the cell type is. So we can definitely help you with that. And what I would recommend is that you contact the scientific support group, and the email and phone was given on that contact slide towards the end of the talk. The email address is scientificsupport@corning.com.
Great. Thanks very much, Marshall. Omar, question for you. Do the no stem cells acquire stem cell markers?
So we think that, at least in the based on our results from the organoid assay, we find that in vivo these non-stem cells acquire the ability to initiate these mini intestines in culture, and once they form these organoids in culture, they then give rise to the LGR5 positive stem cells. Exactly or precisely what is the in vivo identity of these non-stem cells that start to behave like stem cells, we're not sure. We're now addressing that question. But at least in a high-fat diet, these non-stem cells acquire plasticity where they're now able to give rise to cells which eventually give rise to the traditional LGR5 positive intestinal stem cells.
All righty, thank you, Omer. Wojciech, looks like we have one last question for you. One of the audience members asks, for the GFP fluorescence assay of viability, did you use a kit or did you develop your own?
Yeah, this is something I would like to draw people's attention to, because we do not add any external reagents to wells when we measure GFP fluorescence in the spheroids. And you can use some other kits as a backup, but as it is, GFP measurements are very quick, and you can measure the plate of spheroids in 5 minutes, 384 spheroids. So the readout is very quick. You don't add any external kit reagent. So this is why you can reuse the plate and measure it as many times as you want. I think that's it.
Great. Thanks very much, Wojciech. And looks like our last question's for Omar. Omar, was an unsaturated fatty acid control used to see if this is a specific effect of saturated fatty acids?
Yeah, so it turned out that, the high-fat diet is comprised mostly of lard, and the main saturated fatty acid component of lard is palmitic acid, and the main unsaturated component of lard is a fatty acid called oleic acid, which is a mono-unsaturated fatty acid. And indeed, we find that we're able to mimic the effects also with oleic acid. So we don't think that the effects that we're seeing are specific to palmitic acid, but rather to a range of fatty acids, whether they be saturated or unsaturated. And we're currently, probing different fatty acids to put together a better understanding of which ones are able to mimic aspects of the high-fat diet.
All righty. Thank you very much, Omar. And with that, we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be archived on our website for 6 months at www.genengnews.com. So if you missed parts of it or want to watch it again or want to forward it, the link to your friends or colleagues, which we always recommend, you can do that. I'd like to thank Omar, Wojciech, and Marshall again for their informative presentations, and I'd like to thank the audience for their attention and thoughtful questions. And a very special thanks to Corning for sponsoring this webinar. So hopefully we'll see you again at another Gen webinar in the near future. And goodbye for now.